Okay, what we're going to do is we're going to draw the picture that we saw in the PowerPoint, and we're going to represent DNA that is being transcribed into mRNA. So to do this, we're first going to show, so at the top of my paper, I'm first going to draw, this is too big. I'm first going to draw a sequence of DNA that's sort of twisted, and then, whoops, can't see that yet. Sorry.
Wait, I have a question.
What?
Are you talking about right here or like for that? This?
No, either one, but that space is a good space to draw it on your notes. It's fine. Yeah. Okay. So basically, I have a partially twisted strand of DNA that's been opened, and then it starts to retwist. And this will be my 5' end, and then this will be my 3' end. And I'm going to use another color to show the other complementary strand because it's kind of hard to see. So this will be my 3' end, which twists and then is opened and then begins to twist again. So this would be my 3' end, and this would be the 5' end of that strand. And just to show that it's still bound, I'm just going to show some things to represent the nucleotide base pairs, kind of going through it. So that is the DNA before and after transcription. So we learned that transcription begins when the RNA polymerase encounters a promoter region. So on our DNA, it's bigger for the back. All right, so let me know if I start to write off the screen. So on our DNA, and we're going to be reading the strand that starts out as the 3' end. So trace this one here, and this one is where the promoter region will be found. So I'm just going to highlight it as sort of a box. So those nucleotides will represent the promoter region. Okay, so these will represent my promoter region. Also sometimes called Tata box region. Okay, so that's what's represented there. Now at that region, that is where the RNA polymerase will begin to unzip, unwind the DNA. So I'm just going to draw my, so RNA polymerase is going to, I'm going to shade that in a little bit as my RNA polymerase acting around this region here. And it's going to, RNA polymerase is unzipping, unwinding, and it's moving from where the promoter region, and it continues to move in this direction. And this is my RNA polymerase. So let's put some nucleotides in the middle of the open region of our DNA. So basically, in the open region, I'm going to put about 6 nucleotides that we're going to be transcribing here. So 1, 2, 3, 4, 5, 6. And I'm going to name them. So this is going to be G, T, A, G, C, T. And make sure you leave a little space in between because we've got to put some RNA in there. And this side, I'm going to call my template strand. So this is my template strand. And the other strand, which is the one that is not being transcribed, I'm going to put the nucleotide bases on. So this would be CAT, CGA. This side is called the coding strand. So now what's being transcribed is mRNA in this region. So it started with, so the mRNA, as it's being transcribed, it's going to be kind of pushed out to the side. So we're going to draw what the mRNA would be. So this will be my mRNA strand that's being made, and this is the mRNA that has already been transcribed. So this would be the 5' end of the mRNA, and I'm not going to bother at this point to draw the nucleotides because they're there, but I'll draw them here. And so my nucleotides, so remember, so the complement of T would be A, the complement of C would be G, G would be C, and then instead of thymine, what is my nitrogenous base? The U for uracil replaces T for thymine. So there's my difference. Notice the relationship between the coding strand and the mRNA that is transcribed from the template strand. So this is basically the overview of the actual process of transcribing the mRNA to make the mRNA that is called pre-mRNA because it's ready to be processed in order to leave the cell. So in order to do that, let's go ahead and underneath our drawing, are we okay on this drawing before I move on? Would I what? I am going to. Okay. Yeah, I noticed that. Okay. Okay. For those who are absent in mind or body, I will put this on there. Okay. All right. So what we're going to draw under here is we're going to draw what the mRNA looks like before and after processing, a representation of that. So let's just to space it out, I'm going to make a narrow rectangular representation of the mRNA before processing, and then I'm going to have another representation after processing. Okay, so on this, I have the, I'm going to have this will be my 5' end here, and this will be my 3' end. So I'm going to have my cap and my tail, which we'll have down here as well on the final product. And then in between, we are going to have regions. So we'll have some regions that are, I'll have some shaded regions in here. And I'm going to call this shaded region, this is going to represent my exons. Remember, the exons are the segments of mRNA that are going to get expressed. So those are the ones we want to keep. And then I'm going to draw a few of those. And so then I'm also going to have the ones that are not going to be expressed, and those are called my introns. So introns are removed. So then once the introns are removed, then what happens is I am left with the exons spliced together. And this is called the coding segment. And what I have not identified is, so again, this is my 5' end, and this is my 3' end. So on the 5' end, we remember that this is where a methylated cap has formed. That's not very dark. I'll make it darker. And this is called the poly A tail. And that is on the 3' end. So that is something you will always see, if you, in fact, you could see the mRNA. It will be there. So in order for the mRNA to be able to leave the nucleus and be recognized as active mRNA, it needs to have those. Okay, let's review what we've talked about in this drawing, which actually goes over the steps. Okay, so if we start from the top, we have our double helix of DNA. The template strand contains a promoter region, which is recognized by RNA polymerase. RNA polymerase unwinds and unzips and adds the nucleotides for the RNA, starting with the 5' end first. So it adds them in a 5' to 3' direction. And then once that is finished, as it's finished, the DNA will then rezip and recoil after it has already passed. So basically this already has passed, and this is the waiting to be uncoiled and unzipped. And this continues until the termination region is reached. So then once the mRNA leaves, it is capped with on the 5' end, and a tail is added on the 3' end, and then it is further processed, it's spliced. So the parts that we keep are called exons, and the parts that are removed are called introns. And so the introns are removed, the exons are spliced together, and then this is the mRNA that is ready to leave the nucleus. Okay, questions on that?
Once it's been removed, at the end of it?
No, I think that happens before or at the same time as it. So yeah.